My name is Saara Suominen, and today I'm going to tell you about a data management plan that we have developed for a marine invasive species monitoring program that uses environmental DNA sampling in Suva, Fiji. This project is an IOC UNESCO project. It is implemented by IODE and Obst, the Ocean Biodiversity Information System, and it is also an endorsed United Nations Decade of Ocean Science for Sustainable Development project. So our program is called PACMAN, which stands for Pacific Islands Marine Bioinvasions Alert Network. And our objective is to detect invasive species on their arrival in the Pacific Islands, or in this case, Suva, Fiji. Why we're doing this is because increased marine traffic is accelerating the spread of invasive species. These invasive species pose a major threat to the local ecosystem and the livelihoods that depends on this ecosystem. The management of invasive species really depends on how fast these species are detected in the environment. So they need to be detected immediately after they have arrived, before they have become a problem, to be able to really manage and eradicate them. We have a new tool available for us, which is called environmental DNA, where DNA is extracted and analyzed from water samples that are taken in the environment, the target environment, to get a list of species that are present in that environment. Through our project, we aim to develop a monitoring plan as well as a decision support tool so that the environmental managers who are working in these ecosystems can get an indication when an invasive species has arrived in their environment to take the next necessary steps. So why we chose to work with environmental DNA. So environmental DNA sampling is relatively easy, and it also requires less taxonomic expertise compared to traditional monitoring methods. However, it's a very data-intensive process. When you get species information from DNA, it's always derived information, it's not direct observation. This means that there's extensive sample processing in the laboratory as well as on the computer before you can get to your species lists. On the right is a typical workflow used in environmental DNA project. So including field sampling protocols, the molecular laboratory protocols, the sequencing protocols, and finally how these sequences are analyzed to get to your species list. So because this process is so complicated, standardized data management is really key to be able to have reliable and comparable information throughout your sampling program. We also want this information and the workflow to be accessible for all users so that they can easily analyze their data, get their data products, but also understand what is happening throughout the process. We also want our data products to follow the FAIR principles, that they are findable, accessible, interoperable, and reusable. So to get to these objectives, we have devised a data management plan, which has three major parts. It has the plan for data collection, storage, and preservation, a data analysis plan, as well as the plan for data sharing and reuse. In this picture, you can see all the data products that we have identified in this project, from the products that are collected, so the field and sampling information, possible images, as well as the raw sequence data, which are produced by the scientists who are doing the sampling. Then this metadata, together with links to the sequences, is stored in the cloud in RO, so research object crates, which are then published. And in addition, the data will be published in Darwin Core archives on global biodiversity databases like the Ocean Biodiversity Information System. So for the first step, we have prepared standardized field sampling sheets where the user can record the sampling parameters, the sample identifiers, any environmental measurements that were made, any information about images that were taken, and links to the externally hosted sequence data. And all of these are, of course, linked to the sample identifiers that are unique. We will be developing a web interface for submitting this data to make it as easy as possible, and the data standardized formats will make sure that the data can be directly submitted to the analysis pipeline as well. For the data analysis, we have prepared a bioinformatic pipeline. We've called the PACMAN pipeline, and this is based on a compilation of different publicly available pipelines that work on similar projects. So this pipeline basically takes the raw sequences and goes through all the necessary steps, so quality control, trimming of any adapters and primers that are used, inferring the ASVs, which is the amplicon sequence variance. This means that no sequences are clustered, but in fact all unique sequences are retained. So it is easy to go back to, in a way, the raw data. These sequences are then aligned to reference databases, and the taxonomy is assigned based on probability. And finally, and most importantly, in our pipeline, this data with the metadata that is submitted is automatically formatted to Darwin Core Archive format, which then can be easily submitted to the public databases. So very key to our process is that this data analysis can be as automated and as easy to use as possible. And the pipeline is open access. Now, the first version is available in GitHub, but of course it is still under development. And we will also be training the people who are using this pipeline in understanding really what is happening and what are the different choices that are made, and the different parameters needed for each step of the process. So finally, as I mentioned, this data and all the related metadata will be available through Obst. We are still looking into how to deal with any possible sensitive information, and this will be done based on discussions with key stakeholders, but can be, for example, that certain occurrences of, for example, commercially important species or threatened species will not be available immediately after sampling in the public databases. And we will also be developing the decision support tool. So the idea of this is that it will give an indication for the environmental managers whenever a risk species pops up from the data, so that they can go and make further investigations into if there is a risk situation in the environment. And these risk species will be based on invasive species list, but also species distribution models that take into account the species traits, as well as if they possibly could survive in Fiji and become a major problem. So as a conclusion, our goal is to have a working long-term monitoring program that can become routine in risk areas, that is producing data products according to FAIR principles, and especially to empower the local community to continue the development of the project, and that our workflows are clear and easy to use and are throughout producing data products that then can be used easily in the future as well. So for any further information, please visit the Pacman website. You can also find our Pacman monitoring plan there, and in GitHub, as I mentioned, our Pacman bioinformatics pipeline. Thank you very much, and feel free to ask me any questions.